Hello everyone. Today, Pratik and I will be presenting our tool WEPP, which demonstrates that phylogenetic placement achieves near haplotype resolution in wastewater-based epidemiology. So wastewater carries fragments of all the pathogens infecting a population. In wastewater-based epidemiology, wastewater is sequenced to monitor pathogens circulating in a community, and it provides an early warning for infectious disease outbreaks by detecting viral shedding as soon as someone gets infected. Wastewater-based epidemiology offers several advantages over clinical sequencing. First of all, it provides a cost-effective way of estimating a pathogen prevalence in a community. Along with it, it provides an unbiased estimate of prevalence as it does not depend on accessibility to healthcare facilities of all the people living in an area. It detects infected people as soon as they catch an infection without any delay caused by the onset of symptoms. It also provides a non-invasive approach that alleviates concerns about the misuse of an individual's DNA. With all of these advantages given to us by WB, it has some of its own challenges. First of all, it's a very difficult problem to analyze the wastewater data because it's a mixture of multiple genomes which are infecting the population, and most often they have very low genetic diversity, especially if we are trying to analyze a pathogen's variants. It also gets more challenging with the small sequencing reads obtained from the wastewater, and the sequencing errors make the problem more difficult. Due to these challenges, existing tools only focus on estimating lineage abundances, thereby not utilizing the full potential of wastewater. So this graph shows the number of sequences that are deposited every month in the for SARS-CoV-2 in the global database. And we can clearly see that the sequencing rates uh, have tremendously declined. So with this reduced sampling, it gets much harder to detect new variants. And because of this, we now see longer delays in designating new lineages, and such delays in lineage designation directly affect our ability to detect these new variants from the wastewater. So there are a few limitations of existing WBE tools. The first one is that of a limited resolution. So the lineages that are designated, and I'm giving the example of SARS-CoV-2, they have a lot of genetic diversity. So here in the first figure, I have shown an example of lineage BA.1 in this color, which is highlighted in this color green, where two sequences are separated by 50 mutations, which means that even though they're quite different from one another, they cannot be differentiated with the existing tools. The second is that the existing tools use a single signature for all the haplotypes that are within a lineage, and they do not account for diversity within a lineage, resulting in inaccuracies. So here we can see that two of the most widely used tools, how they perform on wastewater samples where we knew the ground truth. And on the x-axis, we can see the expected abundances. And on the y-axis, we can see the estimated abundances. And we can clearly see that the root mean square error of this differences in estimated and expected abundances is non-trivial for both of these tools. The third one is that of a timeliness. So the existing tools are dependent on mutation signatures of designated lineages. And as I mentioned before, with lower sequencing rates, there are delays in designated new lineages, which makes it very hard to discover emerging variants using wastewater. And if a new variant came up, even though we have it in our database, we would still not be able to start searching or analyzing for it in the wastewater due to these associated delays. So, I'll give a quick overview of how WEPP works. So, WEPP requires sequencing reads and a phylogenetic tree in the form of a mutation annotated tree from Usher, which is daily updated to contain all clinical sequences collected till date, and along with their inferred ancestors. So, these mats, they also contain the lineage labels inside them. So WEPP performs phylogenetic placement of wastewater reeds onto this mat to determine not only the lineage proportions of which the existing tools report, but it also estimates the haplotype proportions in the sample from which we are observing these reeds. So in this example, I have shown that there are three haplotype sequences from which we are getting the wastewater reeds, which belong to two lineages, L1 and L2. So WEPP determines both the lineage abundances and the uh, haplotypes and their corresponding abundances. So along with it, uh, WEPP also reports something which is called as unaccounted alleles, which are the alleles which are observed in the wastewater but can't be explained by the selected haplotypes from the mat. And most often we can see because the clinical sequence database might not contain all of the circulating variants due to the undersampling or sequencing delays, the wastewater starts having these new mutations from from these new variants, which can be caught with WEPP now. So, along with it, which WEPP also performs parsimonious mapping of these reads to the selected haplotypes, which allows the user to analyze these reads in the context of detected haplotypes. As shown over here, some reads can map to multiple detected haplotypes, which is most often the case. So WEPP also provides an interactive dashboard to analyze the results better, which will be covered later by Prateek. So this slide captures at a high level of how WEPP differs from the existing WBE tools. Existing tools perform coarse-grained analysis on the wastewater samples by reporting the lineage level trends, while WEPP enables fine-grained analysis of the samples, enabling more insights about the outbreak and unlocking new applications, some of which will be covered in the next few slides. So now I'll give a quick overview of how WEPP works. So as I mentioned before, WEPP requires a mat and a sequencing reads, and it begins the algorithm by placing these reads on the phylogenetic tree. And based on these placement results, WEPP infers the most probable haplotypes for a given wastewater sample. Next, it adds some of the neighboring nodes of each selected haplotype to expand the set of possible haplotypes before estimating their abundances with a deconvolution algorithm based on Freya. We refine the set of haplotypes remaining after the abundance estimation by iteratively adding the neighbors of the remaining haplotypes and computing their abundance till convergence. At the end, when we have obtained the haplotype and their corresponding proportions, we use them to infer the corresponding lineage proportions. WEP also computes the unaccounted alleles based on the final set of haplotypes and also determines the parsimonious mapping of reads to these haplotypes. Okay, now let's quickly take a look at some of the results. So here, what we are showing is the on the same data set for which we had the ground truth, how web performs relative to the other uh, popularly used tools. And here, we can clearly see that the RMSE of web is much better, which is due to the fact that web uh, considers the entire phylogeny of the existing sequences and their inferred ancestors when determining the best set of haplotypes for the given wastewater reads, and the corresponding lineage abundances from these haplotype abundances are much more accurate, and which is can also be explained with a simple fact that a single genome signature for a lineage that has a lot of haplotypes is not sufficient to represent the genetic diversity. Okay, so with this haplotype level resolution, WEPP unlocks a powerful application for tracking intralineage haplotype clusters from wastewater, which so far has been restricted to clinical sequencing. This provides more insights into the state of the outbreak without the bias of clinical sequencing. Here we have shown an example of simulated intralineage haplotype clusters based on the sequences that were observed in San Diego. We can clearly see two haplotype clusters for the lineage EG.5.1.4, shown by the circles of red and green color. We can also see the haplotypes that are detected by WEPP with these yellow colored stars. And similar to this lineage, the other two XBB sublineages also had multiple haplotype clusters, which were accurately identified with WEPP. And in general, WEPP identifies these haplotypes within a mutation distance of one on an average to the haplotype which is present in the wastewater. So this analysis was not possible with the existing WB tools as they would only report the lineage abundance estimates. But with WEPP, we can obtain finer insights about how different haplotype clusters are changing. Okay, so now transmission of haplotype clusters can also be analyzed from the wastewater with WEPP, which overcomes the limitations of clinical sequencing based analysis. WEPP can reliably detect the introduction of these clusters from the wastewater prior to any clinical sequence in the area, which is illustrated with an example where a wastewater sample from Delaware shows the presence of two haplotypes from a small region of the phylogenetic tree, where most of the sequences were collected from California. On seeing such a result, a public health official can infer the presence of a new cluster in Delaware which most likely originated from California, as it was previously seen to be circulating there. So when we compared the result of lineage abundances from wastewater of WEPP and Freya with the clinical sequences found in San Diego, we found that WEPP was much better correlated. And here in this graph, we show this correlation with KL divergence, where lower is better. So we show the we show that even on real wastewater data, WEPP has a much higher accuracy. We also found 32 haplotypes from this data set by the WEPP, which were also clinically observed in San Diego. WEPP also identified multiple distinct Omicron introductions in San Diego from these wastewater samples. So we tested WEPP on wastewater samples from other pathogens like RSV-A and Mpox and found that it performs equally well across pathogens, which demonstrates its pathogen-agnostic nature. So now Prateek will tell us more about the WEP's dashboard and how you can use it to interpret the results better.
Thanks, Pranav. So, the final output of the web pipeline is an interactive dashboard for analyzing the haplotypes detected from the wastewater samples. The dashboard provides a phylogenetic view to explore where the selected haplotypes are located in the phylogenetic tree of millions of clinical sequences, and along with information such as relative abundances of each haplotype and its associated lineage, it helps users to understand how the haplotypes are related to each other and track different haplotype clusters. In addition to that, we also display aligned reads from the wastewater samples mapped to the selected haplotype, allowing fine-grained tracking than traditional lineage-based tools uh, enable. This allows users to dig deeper by inspecting actual reads parsimoniously mapped to these haplotypes. Finally, the dashboard also highlights unaccounted alleles present in the sample but not explained by the selected haplotypes. This could represent novel or cryptic variants, offering an early signal for emerging threats. So, this is the main page of the web dashboard. The, when launched via Web's SnakeMake pipeline, the dashboard loads all the wastewater samples analyzed with Web. It basically loads all the projects from the results folder and displays from here. Clicking on any of the project allows users to interpret and analyze the web results. On the other hand, users can also directly upload all the web generated BAM files and the reference index files which are present in the results folder of a project. So this is the phylogenetic view that comes up upon clicking on any of the project. The left panel lists all the detected haplotypes along with their proportions and corresponding lineages. On the right side, we can see the haplotypes position on the phylogenetic tree. This phylogenetic tree view is useful in analyzing different intralineage haplotype clusters, allowing fine-grained tracking of the outbreak in an area. The left panel from the previous slide gives details about the detected haplotypes, their proportions, as well as the associated lineages. These haplotypes are listed in descending order of their relative proportions by default, which can be changed by the button here. The unaccounted alleles panel show the alleles present in the significant proportions in the sample but unexplained by WEPP's selected haplotypes. Each unaccounted allele is annotated with its residue, allele frequency, sequencing depth, and potential haplotype sources. As mentioned before, these are indicative of novel variants that haven't been clinically detected. Here we want to analyze a haplotype node 3411 in detail. We can click on the zoom button to see it in the context of nearby nodes. This haplotype also has an associated uncertainty, which can be seen by clicking on the uncertain haplotypes. WEPP is uncertain in its detection between the node 3411 and node 34112. We can view this uncertainty on the phylogenetic tree by enabling it with the button. As shown here, we can clearly see both nodes in different colors. To explore further any haplotype in more detail, users can click on the Annotate node, which opens a JBrowse 2 plugin to display the haplotype sequences along with the reads that map parsimoniously to it. The top track in the JBrowse panel shows the haplotype sequence, while the one below shows the parsimoniously mapped reads. Each read is color coded based on mapping quality and mutation presence. This view helps in analyzing mutations of the wastewater reads in the context of the detected haplotype. Further, in the JBrowse panel, to explore unaccounted alleles in more detail, the dashboard provides a filtering menu that only shows the reads carrying a specific unaccounted allele. In the left panel, if you click on one of the allele to filter the reads which contain that allele, the right panel shows that this the reads filtered reads helps us analyze these unaccounted alleles in the context of their potential haplotype sources. Uh, Further, clicking on any of the haplotype sequence opens a detailed panel with key metadata such as the haplotype ID and its sequence along with other details. It shows the list of any uncertain haplotypes associated with the selected haplotype as well as the potential unaccounted alleles that might have originated from here. Now, Pranav will demonstrate a video running the SnakeMap pipeline of the web.
Thanks, Pratik. So before starting the demo, I would like to clear a few things about the data organization, so which you can clearly see by going to the web's wiki page. And here, if we go to the data organization, so if that clearly tells you how the data should be organized. So we go to the web's folder, uh, the users need to create all of their different wastewater samples inside the data folder. For example, here, if you are analyzing the SARS-CoV-2 real wastewater reads, which is a single sample, you create a new folder inside data. And here you put in your FASTQ files, the reference genome, and the MAT. So if and one more thing that you would like to keep in mind is the way in which the FASTQ reads are formatted. So the we expect the FASTQ reads to be in the format like underscore R1 dot FASTQ dot GZ. Similarly for R2. And if you're using a single-ended reads, then they can be like anything dot FASTQ dot GZ. Another thing is for SARS-CoV-2, we provide this reference genome in our repo, which you can copy and put it in this folder. And for all of these different mats that you require, so one way uh, to download these mats is you go to this getting mutation annotated tree, and here, if you click on the first link, these are maintained by the Usher tree. They take you to this page, and here. um you can go, so here in this page, for different pathogens, first you can select the different pathogens like influenza, Mpox, RSV, and SARS-CoV-2. And here in this page, they mention the place. For example, for SARS-CoV-2, this is the link. When you click on this link, you can go and get the public mats. So coming back to our web, in the organizing data. So once you have all of these data, like you can also provide the masking file, which contains the sites which you do not want to consider when you are analyzing the wastewater data. And Optionally, you can also provide a JSNL file, which is used for visualizing the dashboard. If you don't have one, the web pipeline will automatically generate it for you. So once you run any wastewater sample, web generates an intermediate folder for each of the wastewater sample where it keeps some of the intermediate files. And once it is done, you can analyze these results in the results area for your sample. Okay, now I want to move on to the installation part. So there are three different ways of installing. I'll cover the first one, that is how you can install via Docker Hub. So here is my terminal. I created a new directory for this demo, web demo, and as you can see, it is empty. So you just have to copy this. The first step will just directly download this container from web because it might take a little while, but because I have already downloaded it, this was quick. In the next step, you want to access this container. So you just copy this command if you do not have anything in your directory, which is true in my case. Otherwise, if you want to access the files that are there in the current directory, you should use the command below. So another thing that you need to keep in mind is this port. So this port is useful for visualizing the dashboard to your area. So if you want, instead of port 80, if you want to use any other port, you should change this first argument. And you also need to ensure that you have enabled the port forwarding. So I will use this command over here. And I've already run this command in another terminal. And once you run this command, you will see a window like this. And if you are using VS Code, you can do the port forwarding like this. Like you can click on a port and then okay, it's better if I show you. You can just click 80 because you're using a port number 80 and the port forwarding is done. So now you are inside the container. Now you are, okay, you can check if it is properly installed by running this command over here, which uh, prints the help menu over here. That means this container is working fine. So now we are all set to try the examples. So here we have given you two different examples for RSV-A and SARS-CoV-2. And I have already run this RSV-A example. Um, so I'll give you a demonstration of the other one. So before doing that, I want to show you the data organization. So if you go to this data area, there are two folders. One is for RSV-A real and the other one is for SARS-CoV-2 real. And both, if you go inside each of them, the exam, the sorry, the code that we have given, it automatically downloads all of these files that are required. Um, and similarly, I have also downloaded the files for SARS-CoV-2. So here you can easily see the FASTQ files, the reference files, and the MAT as well. So another thing that I wanted to show you is the results. So because I told you I have already run the RSV dataset, so you can clearly see all of these different files, where this file tells you the haplotype abundance that is there. Let me show you how it looks like. Haplotype abundance. Okay, so if you open this file, the first column shows you the haplotypes that were detected. The second one gives you the lineages. The third one gives you the haplotype abundances. Similarly, you can analyze the haplotype uncertainty, which gives you the uncertainty associated with each haplotype that is detected. So in the second haplotype, you can see it is WEPP is uncertain between these two haplotypes. And similarly, you can analyze the rest of the results. Um, so now I will show you a quick demo of this SARS-CoV-2 dataset. As I mentioned before, I have already run all these commands, which will download all of these files within this data folder. So as the next step, all you have to do is copy this next command, which runs the pipeline. Uh, you paste it here and press enter, and it will start running. So it will take a while. I think on 32 cores, we can expect 15 to 20 minutes for this to run. So what I'll do is I'll pause this video, and once we have the results, I'll start explaining you of how to use the dashboard. Okay, so now we have our results with us. The run completed after some time. So now, when web completes, you will see a message like this: the workflow is completed and dashboard is running on localhost 80 or any other forwarded port. So yeah, if you are using localhost 80, you can access it in two ways. Either you can click on this or you can also click on this. If so yeah, let's start clicking on this. Let's see what opens up. Nice. So we have our dashboard view like the one that Prateek explained a few minutes back. So since we have two folders with us, as you can see over here in the results section, wait, what is happening? Oh, because yeah, I have to do this over here because I haven't mounted my current directory. I'm inside my Docker, so I have to go in results. So our results have two projects, RSV Real and SARS-CoV-2 Real, and that is what the dashboard is showing over here. So I'll click on this project and it opens up. So the moment you open up, it shows you the phylogenetic view and it's populating all of these selected haplotypes one by one. And let's wait for a couple of seconds. I think it has done. Oh, now it is done. So as you can see over here, on this left-hand side panel, you see all of the names of all of these haplotypes, the proportions, their corresponding lineages, and you can order them ascendingly or descendingly based on their proportions. Uh, Another thing that I wanted to highlight was, let's say, okay, this is a good example. Here we can see two uncertain haplotypes. So if I click on zoom button over here, it will take me to this particular haplotype. So now if I click on this button, it says show uncertain nodes. So now you saw that web was uncertain about its results between these nodes. So you can click on it again, and then you will only see the initial results. So this is one cool feature. The other cool feature is, okay, first of all, you can always zoom out by clicking on this button. These are the list of unaccounted alleles that we talked about before, which are most often they are indicative of some new variant that has emerged. And if so you get the allele name, you get its residue, you get its allele frequency and the sequencing depth for this at this site. If you click on the possible haplotypes, these are the possible haplotypes from which web feels their this allele is uncertain, unaccounted allele is originating from. So if you click on any one of those, it takes you back to the first panel to that name. So you can try again for a different node. So it will take you to this, and then again you can click on this and then zoom into this. And again, you can see the uncertain nodes by clicking on this. And that's how you can analyze it. Okay, and if you want to analyze the reads that are mapping parsimoniously to this haplotype, you can click on this node. So this is an internal node. This is the same one. You can match the name. And a JBrowse panel opens up. Here, the first panel shows you the gene. The next, this one shows the sequence, and the third one shows you the reads that are being mapped to it. And you can look at different sections of the genome, see what all mutations are present in the reads, what all mutations are present on the haplotype that is selected, and all of these things. So just to give you an example, I have zoomed in. You can see there is an allele A in one of the reads, which is not present over here, and all of these things. And if you want to filter these reads in some way, some order, you can click on these three buttons, and maybe you want to color them based on the number of position each reads map to. So this is what the equally parsimonious position mean, like how many haplotypes to does each read map to parsimoniously. So if you can click on this, you see all of these details about the reads. At the same time, it also shows you the number of possible haplotypes, which is what the EPP number is. So if you count over here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and if you click on the other one, you can count again 3, 4, 5, 6, 7, 8, 9, 10, 11. Oh, there are more. So that is why they are of the different colors. So you can uh, color code them based on this, but by default, they are colored based on the mapping quality, and you can also filter them based on unaccounted alleles, something that Kritik talked about before. And you can play around this to uh, analyze these reads in much more detail. Like here, you can see this mutation is carried by all the reads as well as the haplotypes and so on. So this is about the dashboard. Uh, so coming back to it, that is almost everything about web. And before ending, I would like to thank my advisor, Professor Yatish Turakhia, uh, who gave us the opportunity and all the guidance that was needed in in executing this complicated project. And at the same time, I would like to thank all of our collaborators and the funding agencies who gave us their valuable insights and feedbacks to improve our tool. Heartfelt thanks to everyone. And if you have any questions or any concerns about using web, you can feel free to reach out to us. So that was all about this short demo and the short presentation. Thanks a lot.